Aloha, students. So again, for this Friday, because I'm unavailable to teach at our normal in-person lecture time, I'm recording the video. All right. And hopefully, you found this on YouTube. Right. Let's continue talking about the induced response of our innate immune system. This overview slide takes sort of those flow charts that we looked at in Wednesday's lecture, having to do with comparing the immediate response to the induced response. And I think presents this in a way that hopefully is a little easier to understand. Those flow chart, all the squares on the top, the pink and blue and white, that's what we discussed on Wednesday. Remember, these are two sort of phases of our innate immune system. They all involve pre-assembled, pre-programmed cells and proteins like receptors, as well as free-floating proteins that act as signaling molecules or directly attack pathogens like complement proteins, right? So therefore, all of this stuff, from the moment you were born, you're already pre-programmed to do. We said the immediate response takes care of most things. This is the macrophages and the complement proteins working together so that the moment you get a cut and bacteria get into your tissues, right, these cells and these proteins go at it right away, and you have very good success at clearing the infection within just a few hours, right? So this is, like I said, your typical like paper cut, or if you accidentally got poked by a needle. Right? By the end of the day, you don't even think about it anymore. Right? We said that if a lot of bacteria gets in, like if the wound was a lot bigger, like you wiped out on the skateboard and really scraped up your arm, right? Maybe a lot of bacteria gets in there. And even though this is still common everyday bacteria, right, the resident macrophages and the complement proteins that are in your tissues, maybe they're sort of overwhelmed, right? So after a few hours of them doing their thing, it's still not enough, right? Because after a few hours, the bacteria has a chance to multiply. But during that time, remember that the macrophages, as they are detecting and ingesting phagocytosing bacteria, right, that's triggering the macrophages to release protein messages in the form of cytokines. Right? So certain genes that code for these cytokines, I mean, those genes were already there because they're part of your genome, right? Every cell in your body that has a copy of your genome in it has these cytokine genes in it. It's just most cells never bother to turn these genes on, right? So only immune cells, and in particular, certain types of immune cells like macrophages, when the time is right and they are detecting pathogen, they start turning on these cytokine genes, right? So these genes are transcribed, then translated into cytokine proteins. And then those cytokine proteins, in the cartoons, they're usually showed as these little triangles. They float out, right, as sort of a sign to alert the rest of the immune system that something is happening. If within a few hours, right, the bacterial infection is beaten, right, and the macrophages succeed in eating all the bacteria with the help of the complement protein, blowing the bacteria up also through membrane attack complexes, then what happens to the cytokine levels? They drop, right? Because if the macrophages stop detecting more bacteria, the cytokine levels drop. So maybe at that point, even though cytokine levels in your blood going on in your body were starting to build up, after a few hours, they just subside and go away. And therefore, nothing else happens, right? And so that's why at the end of the day, yeah, you forgot you even had a paper cut or got poked by that pin or that tack, right? If, however, the cytokine messages keep going up because there's more and more bacteria to deal with, now we're talking about the side of your innate immune system that has to keep going for a few days potentially, right? And within that time, the cytokine levels are calling for backup in the form of things like neutrophils that we saw last time, and some other protein components that are expressed by your liver that we'll take a look at today, right? And hopefully those combined components, right, working with the original macrophages and the complement proteins that are still working, right, that together will help beat the infection within a few days, right? And so that's why I say like 99.9% of the time, the infections that we encounter usually result in just being done in a few days, right? And I think we all know we have our fair share of wounds that a couple of days later are getting a little sore and puffy, right? And red and tender and warm. And maybe there's things like pus, right? That's all part of the induced innate immune response. So these lines on the bottom is a different way of visualizing this flow chart to make it a little easier. Let's imagine that on the left here, okay, is the moment you are infected through a cut. Right? And that would be the x-axis would be the amount of time that is elapsing from that moment that the infection started. And on the y-axis, we'll just sort of imagine something that we'll just arbitrarily call response level, right? In other words, this is like the activity of your immune system, right? Let's first look at a scenario where you have an infection, right? And your immediate effector molecules, like things like macrophages and complement proteins, are going to work, right? So your immune response kicks in, right? And it really begins to go to work. But of course, there's only a finite number of macrophages and complement proteins, right, in that area of your skin or your tissues, let's say. But it's okay because that's enough, right? So you have a sustained activity for a few hours, and then what happens, right? The infection is defeated after just a few hours. So what happens to that immediate immune response, right? It tapers off, and then that's it. You're good to go, right? But notice what happens. In the background, you also have this orange dotted line beginning to kind of rise up. This orange dotted line represents, right, the different cells and components that are part of your induced innate response, right? Because in the background, they are beginning to wake up. Because as your macrophages, as part of your immediate response, are releasing things like cytokines, right? Or are cytokines floating around in your blood. So you begin to get in the background some recruitment of neutrophils going on. But the point here is, even though neutrophils start coming in, right, because your immediate response was able to take care of this after, let's say, 4 hours arbitrarily, right? That response, the orange line, also then tapers off, right? Because that's the end of cytokine messages coming out, right? But what happens if the infection persists, that the immediate response is not enough to take care of everything? That means the induced response in the background keeps getting higher and higher and higher as more and more things like neutrophils come in, right? And so now, beyond a few hours into a few days, it is the combination now of your induced response working together with those macrophages and complement proteins that were part of your immediate response, right? The blue dotted line doesn't go away, because it's not like the macrophages and the complement proteins say, all right, whatever, the neutrophils have moved in, we're done, we don't have to do anything else. No, everybody is still working together, right? And hopefully after just a few days, like 4 days, right, that's enough to defeat the infection. And then your immune system, then this response level drops off as you are all better again. Okay. So this way of thinking about it as waves of activity, rather than, I think unfortunately the book often describes this in terms of somehow a switch, right? Like somehow it's a switch. One moment you're switched on to the immediate response, and then when that's not enough, that turns off and you switch on the induced response, right? That's not the way to think about it, because very few physiological, biological processes really function as on and off switches. It's usually waves of activity brought on by changing dynamics of things like signaling molecules and cells being mobilized. And so this is a better way of thinking about it, right? It's just waves of activity. This way of thinking about waves of activity we will carry on later in the semester when we talk about our adaptive side of our immune system. Because as you can probably already imagine, the adaptive side of our immune system would be another set of dotted lines that would start emerging, right, out of these dotted lines that we have on the screen here, beyond the 4-day mark. Okay, so let's go back and revisit our discussion of these cytokines, right? These little protein messages that are released by things like macrophages when they detect that they are encountering pathogens. Right? We introduced this slide last time. We said there are several different types of cytokines, and remember that many of them are responsible for inducing inflammation. Right? And these are the interleukin family of inflammatory cytokines, or IL proteins. Right? Last time we looked at this example, IL-8, otherwise known as CXCL8, and we saw how they are important for recruiting neutrophils. Neutrophils that are released into the blood, they circulate around, they pass by the tissue that's infected. How do they know how to stop? Oh, it's because there are different markers or signals that appear on the inner lining of the blood vessel. The endothelial cells express things like selectins, right, that act as sort of little road signs, right, that let the neutrophil know, hey, you've reached the site of infection, stop, right? And then once the neutrophil is stopped, right, interleukin messages like IL-8 that are coming from the site of infection through the tissue and arriving at the wall of the blood vessel, right, that's what receptors on the surface of the neutrophil detect, right? IL-8 receptors on the neutrophil sort of can sense that, and that changes the cytoskeletal structure of the neutrophil so that it ends up moving in the direction of where these IL-8 signals are coming from. And where are they coming from? Well, they're coming from the macrophages that are somewhere, in the tissue, fighting the bacteria, sending out these cytokine signals. All right, so today let's move on and look at IL-6. IL-6 is another important interleukin. It is one that's responsible for fever, right? Because it induces fat and muscle cells to metabolize, makes heat rise up both locally and eventually systemically in order to make things more challenging for bacteria to really, multiply. It slows down the colonization of the bacteria in your tissues. One of the important induced responses that is carried out by IL-6 signaling is what we call the acute phase response, and this is mediated by your liver. So IL-6, this little green triangle here, right, it's a small cytokine protein that was released from the site of infection, an infection that has now lasted for more than a day, right? So clearly your immune system has to do something more than just let the macrophages and complement proteins take care of it. The IL-6 reaches your liver and it signals your hepatocytes or liver cells to begin to turn on the expression of a whole family of different genes that up until now your liver cells, weren't really caring about, right? So these are proteins that are soluble and then are released by your liver into your blood. Right? And it's a family of about roughly 30 different plasma proteins, right? Many of these increase dramatically, many tens of folds times compared to if you're not infected, right? And the normal generation of these proteins is very low. So a couple of examples we'll look at today of these proteins that are released by the liver as part of the acute phase response, right, are Mannose-binding lectins and C-reactive proteins. So let's take a look at these. First, we'll look at Mannose-binding lectins. We already learned last time that the term lectin describes a protein whose job it is to bind sugars. We already looked at a couple of examples of lectin receptors that might be found on immune cells like macrophages that allow macrophages to detect mannose sugars on the surfaces of pathogens. So here's another example of a lectin molecule that binds mannose. Right? The name is a little unfortunate because it's just called Mannose-binding lectin, right, or MBL, and you say like, well, wait, aren't those other receptors that we looked at also technically Mannose-binding lectins? Yeah, they are, right? But remember, these aren't all discovered by the same people at the same time, right? Because if they were, then they'd be more careful about what to name these things so you don't get confused. Historically, they were discovered at different times, and different groups maybe were not even aware, right, that some other group was already labeling something a lectin that binds mannose. And so, unfortunately, right, this name stuck for this particular protein. MBL, or Mannose-binding lectins. It's a soluble protein, so it's not like a membrane receptor, like those examples we looked at before. It consists of 6 triple helices, right? So in this structure here, these blue things, right, represent 6 separate polypeptide strands, right? Because these are proteins. The part that's sort of a stalk and the branch that comes down is sort of alpha helical in structure. Right? At the end, there's a globular part of the protein, so more of a blob, right, at the end. And these 6 triple helices combined together, they kind of form what looks like an upside-down flower bouquet. Right? Within these strands, these MASP proteins, like MASP1 and MASP2 in green, right, are the right shape to fit in the Mannose-binding lectin. Below, you can see some actual very high resolution zoomed in electron micrographs. So you can see that indeed the shape really does look like this bouquet of flowers. Right? So the MBL detects mannose sugars on the surface of a pathogen. So the mannose sugars here, let's say this is a bacteria, for example, right? The yellow thing, this yellow surface. On the surface are various mannose molecules in red. And you can see the Mannose-binding lectin does a very good job of binding to these sugars. Very nice, strong recognition because it has multiple Mannose-binding sites on it. Okay? It forms a convertase complex. Remember, what's a convertase complex? A convertase complex is a protein complex whose job it is to activate or convert other complement proteins that float by. And in this case, the complement protein is not C3, like we've been looking at before, but this pink egg, right, known as C4. So the C4 binds to the Mannose-binding lectin, and the MASP, the green MASP proteins are proteolytic. They cleave the C4 into C4b, which sticks on the surface now, and the C4a, which can float off and serve as a signaling molecule. Right? This C4b will then recruit another factor, right? And that together will turn C4b into a different version of a C3 convertase. Remember, the C3 convertase that we looked at before was part of the alternative complement activation pathway. Remember, there's 3 main complement activation pathways. The alternative one was the one that we looked at before that uses a C3 convertase, right, to convert C3s. Now you have a C4b, right, that's going to also become a C3 convertase. So it's going to then also associate with another factor and start also converting C3s, right? So this is now part of the classical pathway, right? Or no, I'm sorry, the lectin pathway, right? The lectin pathway that involves the binding of lectins to sugars in order to activate eventually complement C3, right? If you go back to that chart where you have the three different pathways converging down, right, it'll help you place this. Right? What about C-reactive protein? So C-reactive protein is another protein that's made by the liver as part of the acute phase response. C-reactive protein has a similar type of structure as the MBL. You can see here. I'm sorry, here's the C-reactive protein below. The C-reactive protein, right, forms this pentameric structure, right? It has this pentagon kind of shape with multiple polymer subunits, polypeptide subunits, making the sort of pentagon shape, right? It serves as a platform that can bind to the surface of pathogens and acts as a platform that's like a landing pad for this complement protein up here. And this was the complement protein I was saying has a very similar shape to the MBL protein that we looked at on the previous slide, right? So this is another complement protein. This is complement protein C1, right? So if you look at the cartoon on the right, what you'll find is, let's say we again have a pathogen surface like a bacteria. Let us imagine, right, that for some reason, complement protein C3s that are floating around here are having a very bad time, right, opsonizing and attaching to this pathogen surface just by the alternative complement protein pathway, right? It's just not happening very well, right? I mean, if it did happen really well, right, then maybe your immediate immune response could take care of all of this, and we wouldn't even have to be here, right? Because now we're well into the induced response. Right? This was all stuff that your liver had to pitch in order to help out. This C-reactive protein, though, that's been made by the liver now, it binds to the surface because it can bind to phosphorylcholine groups on the surface of the pathogen. And so this C-reactive protein, this sort of pentagon-shaped purple platform, lays down on there, and it becomes the landing pad for complement protein C1. And C1 is a convertase as well. It converts C4 into C4b, and C4b, once again, gets planted on the surface. And once again, C4b will contribute to becoming a C3 convertase. So this was the slide I was asking you to kind of remember to help you keep all of this in mind, right? We said there are 3 pathways to activate complement proteins, right? And in particular, all of them converge on ultimately activating complement protein C3, right? Because at the end of the day, it's the opsonization of the bacteria or the virus particle by C3, right? That's the thing that's going to enable macrophages to be able to have an easier time targeting this bacteria or virus particle, right? Then up until now, right, the macrophage alone wasn't having a very good time of it. So he said the first way that C3 can be activated is by the alternative pathway. Right? That's what we learned about a couple lectures ago. And that results in the assembly of a C3 convertase that consists of a C3b with a BB. Right? And that way that can act on activating C3 molecules that come by. Right? Today we looked at the lectin pathway, right, and the classical pathway that are more considered parts of our induced immune response, innate immune response. Right? So the lectin pathway is the second pathway to act. So Mannose-binding lectins, those upside down bouquets, right, they bind to mannose on the surface of the pathogen, right? And they end up becoming C4 convertases that create C4bs. And so now you have C4bs planted on there, right? So the C4bs then partner up with C2a, right? And together now, this combination of C4b and C2a, this can be thought of now as a C3 convertase, right? Because these combined will also act to convert C3s that are floating by into C3bs to encourage opsonization. The classical pathway is the third to act. It involves the C-reactive proteins that are made by the liver, those purple pentagon-shaped platforms, right? Little staging areas that can stick to the surface of the pathogen and serve as landing pads for complement protein C1. And the C1 ultimately acts as a C4 convertase, again, resulting in the planting of C4b on the surface that then recruits C2a. And just like with the lectin pathway, these two together now act as a C3 convertase, also activating more complement protein C3. You'll also notice that another way that the classical pathway can function is through the activity of antibodies. Because antibodies, if they are present and they also recognize this as being a pathogen, this being the pathogen that the antibodies were generated in the first place in response to, right, then they can also help in this particular pathway to get complement C3 to stick on the surface. But remember, antibodies, you would only have these antibodies if you've already previously had to go through an adaptive immune response in order to fight this pathogen on a previous occasion when you got sick from it and you needed to call upon the SWAT team, right, to take care of it. And that resulted in the production of antibodies that now the next time the pathogen comes along, the antibodies can in fact help, right? So a bit of confusion here because you say, well, if you have these antibodies, then wouldn't the classical pathway actually happen right away as well? Yes, that's true. The presence of antibodies helps the classical pathway activate right away at the same time that your alternative pathway is acting. And that's why, right, the presence of antibodies is so important to have a strong immune response against something like a pathogen that has the potential to make you very sick again, right? But because by definition, you would not even have these antibodies unless you had previously activated your adaptive immune response to make them. That's why we still consider that this is sort of the third or later one to act. Right? All right, let's shift gears a little bit. So when we've been talking about pathogens like bacteria or viruses, right? We are bacteria. Well, let's just say bacteria. Since we've been talking about bacteria, we are talking about things that are outside of the cells of your body, right? The bacteria infect a wound, right? And they're just making their way throughout your tissues, sort of, in between the normal cells of your body. But viruses are different, right? Viruses, they function by getting into your cells, right, and releasing their genetic material, and therefore hijacking the cells of your body and forcing the cells of your body to make more viral particles. So therefore, you can think of viral infection not as being sort of an extracellular type of presence, right, of bacteria being outside of cells, because the bacteria that hang out outside of your cells, they can be targeted by other cells like macrophages, right, or by complement proteins, right? Macrophages and complement proteins, those things don't go into your cells, right? They're just going always in between your cells to try to clear stuff out. Right? But viruses, because it involves the inside of your cells being where the infection is taking place, this is now an intracellular infection. And complement proteins, right, and macrophages are now useless in terms of dealing with something that's happening inside of one of your body's infected cells. So here's a typical virus particle, right? So viruses are just genetic material surrounded by, with some proteins in there, surrounded by a, lipid bilayer, and there might be other proteins on their surface that enable them to stick to and enter host cells. So here is an example of some infected cells down here. Right, these happen to be erythrocytes, red blood cells, and you can see they're being heavily infected by these little virus particles. Right, a couple of things to keep in mind from this picture here. Mature red blood cells don't have a nucleus, right? They are anucleated. And so viral infection of mature erythrocytes might actually be very pointless for the virus particle because there's no real cellular machinery here for the viruses to actually hijack or take advantage of, right? But the viruses don't know that, right? So the whole point is the viruses on their outside have all kinds of receptors that tend to stick to things found on the membranes of our body cells. So this is just a slide that shows you that you can take some red blood cells out very easily because red blood cells are very convenient to take out of the body. And sure enough, you can see that they're covered with these virus particles. The other thing I want to keep in mind, right, is when you look at this image, you realize it's not like one virus particle, right, infects one cell and that's it. One cell can be infected at one time by hundreds of virus particles, all sticking on the outside and wreaking havoc, right? But for simplicity's sake, our cartoons always just use one virus particle, right? Because it just gets the point across. So let's look at this here. Here is a flu virus, right, like the one that's being represented here. And because it has certain proteins on the outside of the right shape to stick to different proteins or different surface features of one of our body cells, that's how they stick, right? And that's how they essentially get in and are able to release the genetic material that then sort of reprograms, if you want to think about it that way, the host cell, right, to favor production now of the different genes that have been brought in by the virus. right? And those genes, of course, code for viral proteins. And so those viral proteins are made, they get processed by the host cell because the host cell doesn't know any better, right? And then everything gets put together as new fully assembled virus particles that then bud off and go off somewhere to infect another cell. And remember, because you potentially have hundreds, maybe even thousands of virus particles, all doing this to one cell, the number of virus particles that are budding off can also be in the thousands, right? And that would eventually end up destroying the host cell. You're essentially using up the resources of the host cell. And now thousands of virus particles can now go off, newly released, to infect other cells. So clearly, our immune system must have evolved to have some system of detecting intracellular viral infection, right? So how do the cells detect this, right? And how do they actually inform or alert our immune system that the nature of the infection is viral, right, and not bacterial? So there's a special group of cytokines that are made in response to viral infection, and these are interferons. Right? A few of these names I think are great, right? Like I like interferons because the reason they were called this is early virus researchers saw that there are certain small cytokine messages that are released by virus infected cells that actually start changing the behavior of other nearby cells that have yet to become infected, right? And those changes that these signals send out are designed to cause surrounding cells to take sort of a defensive steps in trying to prevent themselves from becoming infected by the virus. So in other words, these little proteins interfere with the virus's ability to spread. So they were called interferons. They're special cytokines made by all cells in response to viral infection. In addition, these interferons are also released by natural killer cells, or NK cells, which are a type of white blood cell that we'll take a look at today and also look at more in detail. Right? Interferons are used by natural killer cells to help coordinate and encourage cells like macrophages to really kind of wake up and realize that they need to do a better job of trying to seek out these viruses. There's two types of interferons, type 1 and type 2. Type 1 is a larger family, right? So of different proteins. So when a cell is infected by a virus, type 1 interferons are made. A couple of examples of these type 1 are interferon beta and interferon alpha. So if you look down here, right, here are the ribbon diagrams showing you the structures of these interferon alpha and beta. Again, they're cytokines, so they are proteins, right? So we're looking at the ribbon diagram, the polypeptide chain of amino acids that make up these proteins, and they're small, right? And over here, right, you see the space filling model. So this is what interferon alpha and interferon beta look like, and these are type 1. Right? Type 2 interferons are the ones that are made by natural killer cells to kind of encourage or work with macrophages. Right? And there's only one there's only one example of a type 2 interferon, and that's interferon gamma. So down here, the green one is interferon gamma, right, as the sole example of type 2 interferon. And here's the space filling model. So again, small protein, as all cytokines are, right? But you can tell the interferon gamma is a very different sort of shape than the interferon alpha or beta. So how are viral infections detected by the cell itself that's being, the sort of the victim of this infection? So inside the cell, we have a system. The cell has a system of detecting certain types of molecules, right, that are real hallmarks of viruses. Now, viruses are made of cell membrane, right? So that part doesn't look that different. Certain proteins, yeah, those are kind of different, right? But really what's special about a virus is the way that its genome or genomic material is packaged, right? So many viruses carry their genome as RNA, not just regular RNA, but double-stranded RNA, right? So remember, for those of you who have taken Bio 275, you know this, right? When our cells make mRNA as sort of the product of genes being transcribed and then the RNA being edited, and then the mRNA is shipped out into the cytoplasm of the cell to be translated by our ribosomes into finished proteins, right? That RNA is single-stranded, right? There's normally no reason why there would be double-stranded RNA inside of our body cells. So evolutionarily, hey, here's another example, right? If it's something that you never see inside of a normal healthy cell of your body, it's useful if you evolve to recognize that this is potentially something from a foreign pathogen. So let's look. The virus particle binds to the outer membrane of our cell. It injects its double-stranded RNA. Now, remember, not all viruses are RNA-based, so we're talking about just particularly RNA-based viruses. So many rhinoviruses, that give you colds and things like that are RNA-based. Coronaviruses, right, like SARS, are RNA-based also. Anyhow, inside the cytoplasm of your cell, you're going to have certain proteins made by the cell that bind to and recognize that they're dealing with double-stranded RNA. So, for example, RIG1 and MDA5, these are examples of a family of receptors known as RIG-like receptors, or RLRs. So over here, they're represented by the colorful orange, purples, blue, red structures. You can see the yellow strand that's going between them is the double-stranded RNA that came from the virus, right? So these RIG-like receptors, they fit the double-stranded RNA, and importantly, right, these proteins also fit or buckle into certain proteins that are on the surface of your cell's mitochondria. The surface of the mitochondria has a complex of proteins known as mitochondrial antiviral signaling protein, or MAVs. So that's this big, long gray thing over here. And MAVs is the right shape to bind to these RIG proteins that have been floating around and have managed to kind of clamp themselves on double-stranded RNA from the virus. That ends up bringing the double-stranded RNA of the virus to the mitochondria. Right? And now the mitochondria will actually use this signal to tell the nucleus of the cell, hey, we're detecting the presence of potential viral RNA inside the cell, right? You better go ahead and start turning on different interferon messages to raise the alarm. So how does that happen? In this figure here, again, we have like RIG1, for example, or MDA5, that's going to recognize conformations of nucleic acids that aren't really normally found in our cells. For example, double-stranded RNAs, other types of single-stranded RNAs that have certain modifications on them that aren't modifications that normally our own body cells would make, right? Those can be detected by RIG1. Long double-stranded RNAs tend to be the ones detected by MDA5. In either case, they are brought to the MAVs proteins that are on the surface of mitochondria. And on these MAVs proteins, right, there are other proteins waiting to use this signal. In particular, there's this TBK1 in green here that is associated with this other factor, IRF3. Right? And it results in the phosphorylation of IRF3, right? Meaning when double-stranded RNAs get brought here, the TBK1 phosphorylates the IRF3 protein. The phosphorylated form of that IRF3 floats off, and it activates NF-kappa B, right? And you're like, NF-kappa B, we talked about that. Yes, NF-kappa B is one of these very ancient primitive transcription factor proteins that's very important in our immune response. Right? So NF-kappa B, as I showed you before, is very important for turning on cytokine genes, right? But in this case, because the NF-kappa B is associated with this IRF3 that resulted from specifically viral infection, right? The subset of cytokine genes that the NF-kappa B with the IRF3 together activate preferentially are not just any cytokines, but preferentially the interferon genes, right? And so the cell starts making interferons. like beta or alpha. So the interferon response, right? Oftentimes physicians, clinicians will talk about the interferon response, all right? So that has to do with a whole suite of different physiological cellular changes that take place, right? That are a natural way for your healthy, uninfected cells to be alerted and respond. So here you can see that the three main kind of goals of the interferon response are to induce resistance to viral replication in all cells. So in other words, the cell itself that was infected, right, changes start happening within it as a result of interferon signaling that actually slow down the cellular machinery, right? Because it's the cellular machinery for expressing genes, making proteins, that's actually being used by the virus to make more viruses. So the cell actually stops or slows down its normal machines to kind of try to slow down the production of viruses. That also, part of the interferon response, is it increases the expression, right, of certain types of signals on other cells that make them better targets for natural killer cells, immune cells, natural killer cells, to recognize them and potentially destroy these cells to prevent them from actually making more virus particles. Right? And importantly, yes, right, the presence of interferons are a signal that actually turn on the natural killer cells so that they do go ahead and kill the virus-infected cells. Right? So this cartoon illustrates this principle that a virus-infected cell on the left, okay, detects the presence of viral genome. Here's the virus, right? Here's the genomic material. Maybe it's double-stranded RNA, for example, right? As a result, you get activated into IRF3 from the mitochondria that works with NF-kappa B to activate these genes like IFN beta. So now you make these blue triangles, which is the interferon beta, right? That floats out from the cell. So imagine, right, now you have this cloud of blue triangles coming out from all sides of this cell, right? Much of this is meant to kind of diffuse and float and talk to neighboring uninfected cells that have receptors for the interferon beta on their surface, right? And when these receptors are activated, they stimulate the uninfected cell to start taking some preemptive defensive measures. What types of measures? Well, things like slowing down their metabolism, and they can actually alter the chemical composition of the cell membrane to make it harder for virus particles that are incoming right towards the cell. It prevents them or discourages the virus particles from actually infecting the cell. But because this is a cloud of blue triangles coming out of the infected cell, that means the infected cell itself, right, is subjected to these blue triangles around it. And so not only would you get paracrine signaling, so paracrine is when a signal is sent from one cell and received by another cell, right? You also get autocrine signaling, meaning these blue triangles feed back, right, and are picked up by the type 1 interferon receptors of the infected cell itself. And this basically reinforces or encourages the virus-infected cell, right, to really start taking some steps to slow the infection down. And that might be what results then, for example, in the activation of interferon A expression. So interferon A is now represented as these yellow triangles, right, that float off from the cell. All right. I had mentioned that part of the interferon response is to get cells alerted so that the uninfected cells can protect themselves, right? And the infected cells can sort of change themselves to slow down the virus that's being made inside of them. But we also mentioned natural killer cells play an important role. So what are those? Right? So here, let's go back to this cartoon. We come back to this cartoon a lot during the semester. And I'll remind you that the natural killer cells are these purple ones down here. They actually are made, right, from more of the adaptive side of our immune system, which we haven't talked about before, but functionally, they function as innate effectors. They can still be thought of, right, as cells that already exist in your body, right, and you come sort of pre-programmed knowing how to make these natural killer cells. So these natural killer cells work together with this type of cell, a dendritic cell, and we'll introduce the dendritic cells today, but we will talk a lot about dendritic cells this semester, because even though dendritic cells seem like very lowly cells that don't really do much, they actually are very important for your immune response. So natural killer cells are large lymphocytes that are very quick to respond to viral infection. They have two jobs: destroy infected cells and increase inflammation, right, to encourage a strong immune response. In this data slide over here, it's a very stylized cartoon of a data slide. This is the time interval after infection, number of days. And you can see on the green is the production of interferon alpha and beta, right? So I probably would have made sense for me to point out the yellow kind of curve here is the virus titer, meaning over time, what is the load of virus particles that can be detected in the tissues, right? So within the first few days, the virus titer grows exponentially, right? As the virus is basically able to escape from things like your immediate immune response, right? It's getting into cells, places where macrophages and complement proteins can no longer get to it. But fortunately, you have this production of interferon alpha, right, that already begins to kind of slow the curve, the yellow curve down. Then you have the activity of natural killer cells coming and targeting the infected cells of your body and just killing them to take them out of the picture, right? So that they stop making more virus particles. And then eventually you'll see after, let's say, 5-6 days, that's when you see this red line. And the red line represents your adaptive immune system now kicking in, right? So this is T cells getting involved in recognizing the virus and doing a much more effective job of putting an end to the virus. So that's why the virus titer then really then tapers off after that point. Here are natural killer cells. The way natural killer cells work is rather than targeting a pathogen directly, remember macrophages, neutrophils, right, phagocytic cells like those target the pathogen directly by attaching and eating the pathogen, whether it's a bacteria or a virus, right? Natural killer cells have a different focus. They don't care about directly attacking, let's say, a virus. They care about detecting what cells in your body are harboring the virus. In other words, identifying infected cells of your body and then eliminating those, right? So you can see how that's a very different way of dealing with a pathogen infection. And that's why evolutionarily, right, natural killer cells came along much later than the macrophages or neutrophils. And in fact, they came along as part of our adaptive immune system beginning to evolve, right? And our adaptive cells, right, don't directly attack pathogen through phagocytosis. They are more concerned with recognizing and detecting the cells of your body that show signs of infection. So when these natural killer cells, they synapse, right, or press against another cell, right? And there are many receptors on the NK cell that can detect sort of weird signals that are being presented by the infected cell. And if those weird signals, right, are consistent with a viral infected cell, then the natural killer cell sends all kinds of molecular messages, right, from itself over to the target cell. And these messages stimulate the infected cell to undergo apoptosis. In other words, programmed cell death, right? The cell kills itself to remove itself from the picture. Here is a sort of a time-lapse series showing a natural killer cell, that's what's the smaller one down here, synapsing with or pressing up against a viral infected cell. Okay? The certain receptors on the natural killer cell can tell that this is an infected cell. So the NK cell releases all kinds of specialized messages that are received by the target cell, and the target cell eliminates itself. Right? So you can see within an hour, the target cell has shriveled up, right? And that way it puts an end to its ability to make any more virus particles. The natural killer cell shown here, once this is done, it just moves on, right? And it moves on and it can go ahead and continue its job. So natural killer cells and macrophages work to activate each other. So here you see a natural killer cell. These natural killer cells, it probably doesn't surprise you, right? They are a very powerful way to target and kill cells of your body that are going bad somehow, right? And because the natural killer cells can keep going around synapsing with multiple cells, telling them, okay, you need to die, you need to die, you need to die, you don't want to mess around with natural killer cells, right? Because most of the time, it's better if natural killer cells are sort of inactive, so they don't accidentally cause cell death. Right? So natural killer cells have to be activated. So here's a natural killer cell that's just minding its own business, floating by, right? And it happens to float by where there's this macrophage over here. This macrophage is busy eating virus particles. So it's doing sort of a modest job of eating virus particles, but it's enough to stimulate the macrophage to send out different cytokine signals like IL-12, like CXCL8. Right, that same CXCL8 that recruits neutrophils will also recruit natural killer cells. So the natural killer cell has receptors on its surface that pick up on that, and the natural killer cell makes its way over to the macrophage and it synapses with it, right? And in this case, right, the macrophage has certain molecules on its surface that let the natural killer cell know that, yes, I've attracted you to come press against me here, right? But the point is, I'm not actually an infected cell. I'm one of your friends. I'm one of your teammates here, right? I'm just trying to pass the message on to you that there's an infection going on, and I need you to kind of activate and start going to work, right? So as a result, for example, cytokines like IL-15 activate the natural killer cell. The natural killer cell then starts packaging these chemical messages up in little vesicles inside of it. These are the chemical messages that are going to tell other infected cells that they should die. Also, the natural killer cell begins to multiply, right, in activated form. Okay. And they will go off now and try to find other cells of your body that are indeed infected with this virus that the macrophage is eating up, right? And the natural killer cell will start telling those cells to undergo apoptosis and die, right, to slow the spread of the virus. At the same time, it's sort of a two-way communication here because the natural killer cell makes these orange triangles. These are the interferon gammas, right? And these are received by the macrophage as sort of encouragement that the macrophage really needs to ramp up, right? So like it's telling the macrophage, hey, you're doing a pretty good job here because you let me know about these viruses, but I want to tell you, you have my permission here to really, really start, right, becoming way more phagocytic, ramp up your virus-eating activities, right? And that's what happens. The macrophage becomes more active now, so it can really go out and start trying to phagocytose more viruses. All right, last couple slides for today. Dendritic cells, I told you, were another member of our innate immune system. These cells oftentimes don't get the respect they deserve because dendritic cells really don't do much in the face of pathogens. They don't attack pathogens. They don't detect and tell infected cells to die. What do? Right? Dendritic cells just kind of wander around picking up bits and pieces of stuff, right? And they collect stuff, and their job is to bring that stuff to some places in your body where you have a lot of other immune cells that are ready to kind of check the stuff out and see what the dendritic cells are finding. So DCs sample the environment, they pick up pathogens and products, right? But they don't do anything about them. They're just sort of collecting them and carrying them around. Right? You can see in this illustration on the right here, during an infection, you will also have dendritic cells hanging out with the macrophages. The macrophages are fighting, right? They're fighting the battle. They're eating the bacteria, whereas the dendritic cells are just picking up little bits of stuff, right? These dendritic cells will drain into what's known as our lymphatic system. Our lymphatic system is a series of ducts or channels that drain from our tissues. We will look at the lymphatic system in more depth, but I just wanted to introduce it to you now. Right? So in these drainage channels are, for example, bits and pieces of bacteria, maybe entire complete bacteria, and cells like the dendritic cells that have been busy sort of picking up all these bits and pieces, and now they're being brought to specialized secondary lymphoid tissue structures like our lymph nodes. And the lymph nodes are where things like other immune cells, for example, adaptive immune cells like T cells, B cells, are hanging out. And importantly, these dendritic cells provide a way for these T cells and B cells that are hanging out in your lymph node, potentially physically pretty far away from the actual site of the infection, right? At least this way, these T cells and B cells have a chance to be alerted, right, as part of your adaptive immune response. Okay. And so, in other words, the activity of dendritic cells in the lymph nodes is to wake up your adaptive immune system. As a result, there's an interesting interaction between dendritic cells and macrophages. Right? No, not dendritic cells and macrophages, dendritic cells and natural killer cells. Right? There are three scenarios here. Imagine, if you will, right, that these dendritic cells have brought different pieces of pathogen into a lymph node. Okay? And in this case, there are some inactive natural killer cells just wandering around in there. Right? You can tell they're inactive because they haven't yet made their little sort of specks or little dots inside of them. Right? So if this is sort of the balance, you have lots of dendritic cells that are carrying evidence of pathogen, right, like virus particles, and you have some natural killer cells that are not activated, then the dendritic cells will help in the activation of the natural killer cells, resulting in the natural killer cells turning on, right? They're starting to make all their spots inside of them, right? And they're activating so they can then go off now and try to find other cells in your body that were infected with whatever the stuff is that the dendritic cell brought in. Let's look at the second scenario. Imagine if the dendritic cell carrying this stuff enters a lymph node that already has a lot of activated natural killer cells already in there. In this case, something kind of weird happens. Right? Then the natural killer cells associate with the dendritic cell, and the NK cells kill the dendritic cell. It tells the dendritic cell to undergo apoptosis. Why? Right? What a sad fate to this dendritic cell that was just doing its job, right? Because what type of scenario would this be? If you already have a lot of activated natural killer cells, this probably means that your induced innate immune system, or the induced phase of your innate immune system, is already well on its way to battling the infection, right? And you might not need or want to really wake up the full-blown SWAT team, the adaptive immune response, right? You might want to see if the induced immune response is enough to take care of it. So that's why, if you have a lot of activated natural killer cells, right, this is the immune system's way of discouraging the dendritic cells from actually waking up the SWAT team, right, the adaptive response. On the other hand, the third scenario here is if you have lots of dendritic cells bringing in evidence of virus particles and very few activated NK cells, the NK cells tell the dendritic cells, yeah, right, you better go out and wake up the SWAT team because we're in a load of trouble here if there's very few of us activated natural killer cells and a ton of evidence that there are viruses all over in the tissues, right? And in that case, the dendritic cells go off and they wake up the adaptive immune response. Right? And so in this image over here, very cool microscopic image, you have a lymphocyte like a natural killer cell synapsing with a dendritic cell, right? And who knows what interesting conversation they are having here and what's going to happen next, right? All right, so we will learn more about this in terms of the role of dendritic cells and NK cells as we move on and talk about things like our adaptive immune response. But for today, that sort of wraps up our discussion of our innate responses. Okay. All right. So I won't see you on Monday because it's a holiday. So I'm not going to see you until next Wednesday. All right. But hopefully this lecture is informative. If you have any questions, shoot me emails. I hope all of you have a great weekend. I'll see you next week. All right. Bye-bye.